So hello, everybody. My name is Justin Toll, president at Aduco. You're in the right place at the right time for a special presentation today. So welcome. We have about an hour and 30 minutes. And this is also has a very special component with a live Q&A for those that are attending this live at the end of our program today. We have this special opportunity to get questions going from clinicians to the man himself. Dr. Chris Shade is going to be joining us, and he's with us today. So, so let's forward the slide, if you don't mind. Okay, so welcome. Integrating detoxification, metabolic fitness, and mitochondrial health for vital immunity and longevity. I'm interested in that. This is presented by Dr. Christopher Shade, and we're going to go about 30 minutes. Now, we're in a special webinar setting. There will be people that are attending live right now with us, and we'll also have this as a part of our archive of valuable information. So if you are with us live, there's a little area down below your screen here that is called chat. And you can chat to us as the panelist, questions as they come up, whether you're thinking for yourself or in clinic, talking about these types of products and using them for immunity and longevity, mitochondrial health, metabolic fitness, and detoxification. And so put those in there as we go along, and we'll see how many we can get to at the end of that. Okay. So, so first off, this content of this production is intended solely for healthcare practitioners and educational purposes only. The information set forth herein is holistic in nature and not offered as a complete or comprehensive training on all subjects referenced, has not been evaluated by the Food and Drug Administration or any other medical body. It is not intended to be a substitute for professional medical advice, diagnosis, or treatment. We offer this information with the expectation that practitioners shall use their own discretion, perform their own research, and rely on their own clinical experience and practice according to the scope of their practice and the limits of their license. Whew. I had to take 3 breaths to say that. Yeah.
That was a lot.
It was. If you go one more, Dr. Shade. So many of you know Dr. Shade before. He's the founder and CEO of Quicksilver Scientific, basically a pinnacle in the world of the best of natural medicine and these products. He's been working to uncover the curative power of nature, basically, and having many decades in our industry, really even coming from a place of understanding farming and agriculture in the beginning, and also the toxic effects of heavy metals and mercury poisoning. And, very eclectically, being curious and, an incredible background in education, developing an understanding of evaluating toxins, and then how to detox ourselves and how to get nutrients into our bodies. And they have at Quicksilver Scientific the most innovative way in the world for delivering nutrients and other pieces towards health and to activate our systems for longevity, health, aging, all kinds of great things. And so we have a really special program here. We have Dr. Shade with us in person. I just want to say how much I appreciate you and welcome to all of this. And so welcome, Dr. Shade.
Thank you very much. I'm happy to be here, and I'm happy to talk to the Aduco Forum again. I had a great time coming out there and talking in person. So I'm going to start this by going back to this front slide and give a kind of an idea of where we're going to go and what we're trying to get to. One of the things you have to sort of get around, we have a very different technological approach than a lot of your practitioners are used to, being standard process people. Standard process, you're looking for this whole plant, lots, there's some main drivers and a lot of cofactors around it. And, I grew up I didn't grow up in organic farming. I grew up being a reductionist scientist in an academic household, and then had this explosion of awareness and consciousness and went into organic farming and was very much in the whole food space. It's kind of a circuitous path for me, but I ended up looking at environmental science and pollution issues, and then getting deep into understanding biochemistry around toxins. And that's where I got to sort of understand more about different triggers that the body has, where you can hit one thing that activates a whole cascading response. And as I then got into the nutraceutical industry, I started getting more comfortable with using single compounds out of a plant that activate whole body responses through what are sometimes called nuclear transcription factors. But you're activating a complex response, even though you're coming in with less broad of an array of chemicals. And now, one of the reasons that you needed a whole plant approach before was that the bioavailability of the key actors in these was pretty low, and getting a broad range of compounds together often quite helped or quite augmented the bioavailability of the main drivers in there. So as we go to a platform like this that's based on using lipid nanoparticles. Now that can sound scary, but the lipids that we're using are mostly things that make up your cell membrane. And when we say nano, that just means that they're very small. They're below 100 nanometers. And people hear a lot of bad things around nano. I mean, it could be things like mineral nanoparticles, like titanium dioxides, get into your cells and they're free radical generators. And this is true, and this is a bad thing. But in the lipid nanoparticle field, you actually are making lipid nanoparticles 24/7. As you take dietary fats, you pull them into the enterocytes lining your GI wall, and your endoplasmic reticulum wraps those fats in a layer of phosphatidylcholine, the same cell membrane phospholipids that we're using to make our delivery. And it's releasing those into the blood to bring dietary fats to the blood. And these particles are down as low as 70 nanometers, making them nanoparticles. And so this lipid nanoparticle distribution of things into the body is quite endogenous. And thus, these particle systems that we make are quite biocompatible. And of course, you've got enzymes throughout the body to take these nanoparticles apart and use the different components. So we're using a very biocompatible delivery system to take things like resveratrol or berberine, or sometimes whole plant extracts. Like in our bitters, we're using whole plant extracts of myrrh and gentian and solidago and dandelion. But in this context of the delivery system, we get more in, and we can activate the biochemistry more powerful with a lot less. And often we are focusing all this absorption into a short amount of time, 30 minutes, and all these things peak in the blood. And we're able to activate a lot of the detoxification or cardiometabolic pathways very rapidly because of this uptake. So the bridge there is we're activating the body's own systems. We're not coming in like a drug, but even drugs are trying to do that, but they're not biocompatible, and they're not bringing an array of things together. Now, it's very rare you'll see a single compound in use in our protocols. We'll bring multiple because, in favor of getting a broader approach. But we are using the body's own systems to do this healing. So in this talk, we're going to be talking about integrating together detoxification protocols, metabolic fitness protocols, and mitochondrial health protocols. And I say protocols because we're usually bringing together multiple different products to hit all of the body's responses. So you see me deconstruct the whole food into, well, actually one compound can do it, but then I reconstruct into bringing in multiple compounds to really round out the whole body approach. We're not saying this pill with one thing in it is going to do everything. We are putting together protocols. And you're going to see detoxification, metabolic fitness. What do we mean by that? So metabolic health is really your body's ability to burn things, the carbon inputs, whether they're fats or carbohydrates or the proteins. How effectively do we burn them versus storing them? And in the sort of everything we've been doing since the 80s in the food pyramid, we have too much carb, too much sugar, and that drives processes in the body that store resources rather than burn them. And that leads to fatty deposition, plaque depositions. And with the fatty depositions comes the release of the chronic slow burn, the NF-kappa beta type slow inflammation that causes a lot of metabolic problems. So you see a metabolic input problem turning into a inflammatory response problem, turning into more chronic degenerative problems. So enabling our body to flip back and forth between burning carbs and burning fat to burning everything very at an optimal level to minimizing fatty deposits and using resources right away is what we call metabolic fitness. Now that overlaps with mitochondrial health, and we're going to see how all these overlap for, as it says here, vital immunity and longevity. So we're going to relate toxin accumulation to dysfunction in immunity, and then we're going to relate it also to dysfunction in longevity programs. So we're going to see this overlap between all those, and then in metabolic and mitochondrial, we'll see all that. So these are the themes that we're going to see keep coming up left and right. One of our star players in this is NAD. We see a picture of NAD and all of its functions in the body. So let's get into it now. Now, what I want you to see is this overlapping function between detoxification and AMPK. AMPK is a trigger that is activating this metabolic fitness aspect, this ability. So AMPK, the AMP kinase, is what is activated when we do carb restriction, keto diets, heavy exercise, and that's activating our ability to burn down all the stored resources and have metabolic fitness. And then over there, we have mitochondrial health. And we'll see that all of these, let me just get a laser pointer here. All of these are overlapping and turning towards one another. Now, one of the key transcription factors, one of the key triggers that activates a lot of things is called NRF2. And NRF2 is going to activate a lot of our different detoxification programs, but it'll also be part of activating metabolic fitness and part of ensuring mitochondrial health. All right, so now looking at detoxification, glutathione, which is core to detoxification and immune response. And then we'll flip and we'll look at it towards longevity. So anything that decreases glutathione, and all of the toxins have a hand in decreasing glutathione. Many of them directly, but because you have to conjugate glutathione to the toxin to get it out of the body. So any of them that do this are going to disrupt the immune system. And here we're looking at endocrine disruptors that deplete glutathione by draining down glutathione as you detoxify it. In antigen presenting cells, what those are immune cells, promote what's called a Th2 polarization. So immune system, something comes in, some invader bacteria comes in. The first thing that you do, you want a Th1 response, kind of actively going out and killing it. Secondarily, you'll form an antibody response. Now, Th2 is more type of an allergic type of an antibody response. So now you're encouraging your body under low glutathione levels to go into this Th2 response, which is a lot of excess inflammatory response. It's not very well targeted to the invader coming in. Now, one of the things that happens, and this is timely right now, is there's a movement of inflammatory response into the lungs. And it's sort of an unbridled, unfocused inflammatory response. So whenever your glutathione is low and you have some sort of infection, you're going to be prone to moving a non-targeted inflammatory response towards the lungs. So we don't want to do that. We want to keep toxin levels down. We want to keep glutathione levels up to able to maximize your body's response. Now, towards longevity, glutathione is a major driver of longevity. In fact, it's been shown for a long time that the longest lived people have higher levels of glutathione. Now, if we look mechanistically, one of the things that's involved in longevity response is telomerase. So telomerase is an enzyme that lengthens the ends of the telomeres. Every time you reproduce your genes, the ends of your genes, which are a buffer part, so sort of something you can grab on and stretch them out and start reproducing the gene. These handles on the end get shorter and shorter. And as those get shorter and shorter, the integrity of the genome goes down. Now, in the reductionist science, they're having a hard time kind of saying why that is, but they've definitely measured the shorter the telomeres, the less metabolic fitness you have, the shorter lived the organism is, and the less wellness or health span the organism is. Now, I would speculate that this is about the sort of holographic activity of the DNA to, sort of project what its different potentials are. And as that end shortened, there's less of your own inner potential that you're able to project, and you have to fall down on default mechanisms. So it has been shown that the more telomerase activity is, the longer these ends can stay, and the more robust the biochemistry can be. And turns out that the telomerase activity is correlated with the gene activity. And so this was in a cell culture, and after a stress to the genome, a stress to the cell culture, you see that glutathione goes down and telomerase activity goes down with it. On the flip side, glutathione in your body is in two pools, the reduced and the oxidized. You want very, very little oxidized glutathione. You should be on this sort of 1% oxidized, sort of 100 to 1 reduced versus oxidized. And in this graph over here, as we stress the glutathione pool and oxidize it, have equal amounts of oxidized and reduced glutathione, you have low telomerase activity. And as you more reduce the glutathione and more reduce down to this roughly 1% oxidized, you have the highest activity of telomerase. And so this reduction potential in the cell is related to the longevity of the cell. So what are the things that oxidize glutathione? Well, having your whole ability of the whole chemoprotective system down leads to more oxidative stress. And we'll see that's related to NRF2 upregulation, which is part of detoxification. And it also relates to the activity of NADPH. So in the NAD pool, which is the central driver of your mitochondrial function, your NAD is partitioning between NAD, which should be in the oxidized state for oxidizing carbon substrates, and NADPH, which is the sister molecule, which should be in the reduced state and drives reductive chemistry. And we'll look at that a little bit later. But the NADPH in the reduced state is responsible for re-reducing oxidized NAD. So we see this overlap between NAD and the mitochondrial pools and its ability to create reduction in the glutathione system. AMPK, that cardiometabolic trigger, we're going to see is crucial in the next slides to xenophagy. When you activate AMPK and you start burning all of your fats and burning all of your glycogen, you also activate something called autophagy, which is the inner eating, the pruning of old cells. And we're going to see that results there's a part of autophagy that is responsible for antibody formation, but AMPK is also responsible. It's a big part of the inflammatory balance in the body, and a lot of this is through the renin-angiotensin system. So in COVID infections, the renin-angiotensin system is a big target of that. The H2 receptor that COVID comes in through is part of the renin-angiotensin system. It's divided into ACE and ACE2. ACE is a pro-inflammatory, drives higher blood pressure. It's invoked when you have a lot of blood loss. Like if you're injured and losing blood, it'll tighten things up. slow down blood flow and keep up blood pressure. Now, ACE2 is responsible for relaxing that and decreasing blood pressure and decreasing inflammatory cytokines. So they need to be balanced. And in these infective models, a lot of that damage to the ACE2 receptor is part of the pro-inflammatory things that are going on there. So you need a good balancer of ACE1 and ACE2, and that's part of what AMPK does. Now, when you get down into its response to invaders, you go back to this idea of autophagy, and autophagy is really important for pruning away old cells. Now, there are several subsets of autophagy. Mitophagy is pruning away old degraded mitochondria. Now, when we talk later about membranes, how do you know if you're going to go into autophagy and mitophagy is going to be activated and you're going to degrade old mitochondria and then make new ones? How do you know a good mitochondria from a bad mitochondria? Well, it turns out that the membrane polarization around the mitochondria, and if you know pictures of mitochondria, they've got all this folded up membrane on the inside, the inner membrane, and they got a round membrane on the outside or elliptical. That's the outer membrane. The strength of the mitochondria is dependent on the polarity of the membrane, or the polarity of the membrane drives the enzymatic reactions and drives the activity of the transporters that are moving things in and out of the membrane and drives the activity of the electron transport chain that's making ATP. Now, as mitochondria get degraded, they have they accumulate markers on their membrane that say that they've got a weakened polarity. And then those markers are like, saying, hey, here I am, I'm a bad mitochondria. And when you go into mitophagy, you look for the mitochondria with the markers on it and you degrade them and then make new ones. Now, lipophagy is part of autophagy, and that's burning away stored fat and fatty deposits. And you don't want to build up too much fat because that's where the inflammatory burns, the NF-kappa beta, that chronic inflammation, those markers come from. Things like endoreticulophagy, say that 10 times over and over. That's for breaking down old damaged endoplasmic reticulum. Endoplasmic reticulum is just a big folded over membrane that catalyzes a lot of the biochemistry. And so if they can't polarize, they can't work. And then when you look at like a whole immune cell, the polarity of the cells, what leads to encounters with a microbe and then engulfing that microbe. And when you don't have polarity, you can't do that. So when polarity is lost on membranes, they get tagged for going into the recycling bin. Now, the last thing, xenophagy, is degrading microbes. So just like, and here you have autophagy in immunity and inflammation, and just like you need to take, in fact, let's go back to those pictures. So in autophagy, you start forming an isolation membrane. Here we have a picture of a mitochondria. We imagine that's been tagged for having a poor membrane. It gets pulled into this vesicle. The vesicle closes around the mitochondria, and the lysosome, which is another vesicle, and these vesicles look a lot like the liposomes that we make for delivery, but they're inside the cell. Now, the autophagosome joins with the lysosome and forms what's called a autolysosome, and in the lysosome, you have various acids and enzymes for breaking all of that down and recycling it. Now, when we go into immunity, the same thing happens. We take a viral particle or a bacteria, we pull it into the cell, we encase it in like a autophagosome, but it's autoxenosome, and it's for taking the viruses, bacteria, and parasites and joining them with a lysosome, and boom, you have an autolysosome, just like you were going to do for the mitochondria. But now you're going to break down this pathogen, and you are going to take parts of the membranes, and you are going to read the chemistry on the outside of the membranes, and you're going to create antibodies to it. So this is part of the deeper immunological response to infections, in that you're going to create an antibody response. So that's all dependent on autophagy and your ability to go back and forth between what's called mTOR forward and mTOR blocked. mTOR forward is laying down of mass. mTOR blocked is going inward and stimulating autophagy. So your ability, and that's all driven by AMPK activation. AMPK activation lets you go inward. So your ability to toggle back and forth between those two viewpoints of your biochemistry is part of your robustness of your immune response. And so the more that we can do these cardiometabolic interventions, the easier we can toggle back and forth, and the more robust your immune response. Let's just skip through that one. Now, the mitochondria are generating all the energy to do all of this work. And here we see the mitochondria as the central hub of the immune system. So the ability to create energy to run these different metabolic programs, or here run these different immunological programs, is at the core of our ability to run high-level immunity. And in this paper, they start talking about chronic energetic stress leading to the immunosenescence. So senescence is when things get old and stop growing, they go into senescence. And so in anti-aging, there's getting rid of senescent cells, but immunosenescence is the aging of the immune system. So you can look at it like this way. When you're young, or when you have a vital mitochondrial energy and you can generate a lot of power, and something comes in and you get an infection, you run it like a really high-grade war. You've got bombers, you've got boats, you've got troops, you've got special forces parachuting in, you've got CIA on the ground assassinating people. And I'm sorry, that sounds, non-pacifistic, but that is what goes on in your body when you're doing this, and you wipe out this invader. Now, when you're going into immunosenescence and you downgraded your energy put into your immune response, now it's more like you're going to make a pot of coffee, you're going to offer the invader some coffee, and you're going to plead for whether it'll give you some mercy. So you don't have a lot of options on the table except a cookie and a cup of coffee. So it's a very different approach. So the more energy you have, the more you can go into a high-level approach. Now, the mitochondria and aging, this has been known forever. They've had different theories on it. It was a free radical theory. We'll look at one that deals with more around NAD and communication between mitochondria and the nucleus, and the ability to keep high mitochondrial strength as key to longevity. But it's long been known, whatever the mechanism, that more mitochondria, more ATP, more longevity. And you remember me saying more glutathione, more longevity. Turns out glutathione is one of the most important antioxidants inside the mitochondria. And how do we make a lot of mitochondria? How do we keep mitochondria up and healthy? Through AMPK activation and consequent sirtuin activation, leading to another trigger called PGC1 alpha, which makes your mitochondria. Now we can go backward to this picture here, and it's basically saying that for long-term metabolic fitness, you need to activate PGC1 alpha to make more mitochondria. And part of that is AMPK activation, and the other part of that is sirtuin activation. And those two go together to activate PGC1 alpha and make more mitochondrial density. So turns out you can have more mitochondria in your cell or less mitochondria in your cell. So you want to clear away old ones through AMPK activation or autophagy, and then add on sirtuin activation to get mitochondrial density through mitochondrial biogenesis, which comes from that trigger PGC1 alpha. All right, so now we're going to speed up a little bit and go through talking about detoxification. We'll talk about cellular detoxification and systemic detoxification, and then the crux between them all, the big driver, the control system and everything, and that's the liver. And the liver has different options, and we call it liver directionality. And from the blood to the liver, to the bile, to the GI, to the stool is the direction we want. Actually, broader than that, it's from the cell to the blood. That's the cellular aspect of detoxification. Then you're in the blood. Now we want to filter with liver and kidneys, dominantly liver, pull from the blood to the liver, from the liver through into the bile tract, and dump down into the GI. And then we got to make sure that we catch everything there with binders and take it all out. So that's liver directionality. And we'll see, unfortunately, there are interrupters of that. different inflammagens, things that generate inflammation, different autonomic states. Are you in fight or flight? Or are you in rest, digest, repair, regenerate, detoxify? Do you have hormone balance? Are you estrogen dominant? All these things are ways you'll see that stress blocks liver. Whether stress cascades down to liver or toxicity cascades up and creates neurological stress, you can't decouple the brain from the liver. And when they are in order, your cells are detoxifying, moving to the blood, moving through the liver, moving to the GI, and moving out. So we'll talk about toggling back and forth between drainage or things that block drainage. Then we'll switch over to AMPK and metabolism. And right before we get there, we'll say, oh, well, wait, a key driver of whether the liver is moving and draining right, and whether the liver is rebuilding its architecture is this AMPK, this thing that we thought of with carb restriction, fasting, keto, exercise. It's actually core for liver regeneration. So we'll talk about AMP and metabolism. It's also core for sealing up things like leaky gut. We'll talk about that importance. Then we'll get into NAD and mitochondria, sort of icing on the cake to drive everything. And we'll see how far NAD reaches into even making glutathione happen, making glutathione regenerate, enabling you to detoxify. So we'll see how all this stuff wraps together, and the crux of it, the sort of pinnacle of it, is having high NAD levels. All right, so going back to a cellular level of detoxification. At a cellular level of detoxification, I often call this pushing away. You're a cell and you're pushing things out of the cell. And I use this paper that was scanning through various zebrafish cell lines. They'll take these cells that reproduce rapidly, mutate them, and they'll have hundreds of different cell lines. And they'll scan them for different abilities, like what's your superpower? And this cell's superpower is the ability to resist cadmium. Now, this brings us up to a language differential too. In toxicity, people used to talk about load, like you have an ability to handle a certain amount of toxins, and you go beyond that and you're super sick. But turns out that that level at which you get sick is very dynamic. So we want to talk about resistance. That's when your level at which you get sick is really high, and susceptibility is when the level at which you get sick is really low. But that level, when that level's high, it's ability of your body to be kicking things out. So when that level's high, you're also lowering the levels in your body. You're depurating, you're detoxifying, you're moving it out. And when your resistance is low, that means the whole chemistry that gets rid of things is low. So you're a net accumulator at the same time. So in your body, you're a net accumulator, and at your cell level, you're very susceptible to what's in there. Again, when your resistance is high, then you have high resistance, low susceptibility. That means the rate at which your cells and your body get rid of things are really high anyway, so you're going to keep a low toxin level. So this is what we want to promote, not just hide from the toxins, but promote our ability to get rid of them. So anyways, in the cell, they found cells that were resistant to cadmium and also cross-resistant to mercury and arsenic. That's important. Those three go together. Mercury, cadmium, and arsenic has the same detoxification mechanisms. And in these cells, they could deal with a lot of these at a really high level. And imagine your Petri dish, and you fix the body burden there at 5 millimolar. It's a concentration, and the cells are fine. And then they go into the cells. They say, why are the cells fine with this? And they had three things. They had high glutathione synthesis rates. They had high activity of a conjugating enzyme called glutathionase transferase, and a high level of a transporter that's called an MRP, or multi-drug resistance protein. So in the cell, the metal's bound to a protein. You've got glutathione. You have a transfer protein that pulls the metal off the cellular protein onto the glutathione. Now you have a complex, and you have a transporter that pushes it out. That's pushing away. Now, if they went in and slowed down any of that, all of a sudden the metal stays in the cell and the cells die. So the cells lose their resistance, they gain susceptibility, they die. You bring that level back up, they live. So the body burden didn't change. What changed is the ability of the cells to deal with that. All right. So we want to encourage that to push things out of our cells. And it turns out when we get down to NRF2, when we trigger NRF2, we trigger all these mechanisms to push out of the cell. So this is a key part to detoxification, is pushing out of the cell. But if we just push out of the cell and we don't have the processing organs, liver and kidney and GI, actively moving out of the blood, we're just going to grow our blood burden and move into things like the brain. We're going to redistribute. These are the old problems in detoxification before, were redistribution of toxins, where you push from the tissues, but you don't get out. And so you just move it somewhere else. And that's all of the symptoms of detoxification. Remember, I said the liver has two directions. It can go into the bile. The other direction is it can accumulate and then can't get to the bile because those transporters are blocked, and then it opens transporters back into the blood, dumps into the blood. You have this back flush into the blood, and that catalyzes redistribution, and that catalyzes all the symptoms, the negative symptoms of detoxification. These are things like lower back pain as the kidneys try to keep up the slack, and then they get all the stress on the kidneys. They fail then you get toxins coming through the skin. You have itchiness first. That's toxins and bile salts itch on your skin. So if you ever itch, you probably have a little bile blockage. And then you're rashing out from the immunological reactions. You have these things going to your brain fog get worse. You're all exhausted. You're blowing out your thyroid. You're blowing out your adrenals. You're blowing out your mitochondria. And you go, oh, that's a Herxheimer reaction. No, that's a failed detoxification. So we need the cellular level, but we need it coupled to the systemic level. So the cellular level, I talked about glutathione, glutathionase transferase, and the transporters, but they're all there within this whole chemoprotection system. I used to call it the antioxidant detoxification protein repair system, but it's the chemoprotection, protecting you from the chemo. That's not just chemotherapy, but this is free radical stress, which is your own internal chemical stress. And it's the metals and the pesticides and the herbicides, this chemical stress, things called electrophiles, which are oxidizing your cell. And this is a big integrated system. And we used to think of these things in the middle as the heroes of the system, but really these are the nouns, the antioxidants, glutathione, lipoic acid, thyroidoxin, vitamin C and E, and CoQ10. All these get activated through the verbs here, the enzymes that take them into their respective functions in their different compartments in the body. So when glutathione is going to quench a lipid peroxide, it's going to do that by donating its electrons through the agency of glutathione peroxidase. And in doing that, it is then going to create oxidized glutathione. Remember, we said oxidized glutathione is no good. We don't want it to build up. It shuts down different luxury programs in the cell and puts you into default scared. It's sort of like fight or flight for the cell. So we need to re-reduce that and bring it back into our reduced pool. And we do that through the reductases. Glutathione reductase. If it's thyroidoxin, it goes through thyroidoxin peroxidase, gets re-reduced through thyroidoxin reductase. Now we can use the electrons from glutathione to re-reduce oxidatively damaged proteins through glutaredoxin. Again, oxidizing glutathione, bringing it back through glutathione reductase. We can use it for detoxification, go through glutathionase transferase and through the transporters. So these are all of the activities that are brought out. And fortunately, there is a way to upregulate all this at once, and that's through NRF2. Now, detoxification is usually spoken of in phases. Phase 1, an activation. Phase 2, conjugation. Phase 3, transport. Now, yeah, we don't go into this. Phase 1, this activation, we need this for non-reactive chemicals. So this would be like PCBs, which were built to be non-reactive, flame retardants, which were built to be non-reactive. But we also need it for a lot of pesticides and herbicides. And what it does is it creates this little radical-like thing, a real reactive molecule, which then in phase 2, we can link on one of our detoxifying compounds to. Now, there's 3 main ones. Glutathione's one, glucuronic acid is one, and sulfate is one. And you have conjugation enzymes for each of those in phase 2. Glutathionase transferase, glucuronosyl transferase, and sulfotransferase. And so depending on the chemical, you use sulfate, glucuronic acid, or glutathione. So that's all phase 2. And in phase 2, you take, maybe it was fat soluble, now it's water soluble. Even if it was vaguely water soluble before, it's now in a conjugate that has a certain charge and a certain shape so that it's recognized by the transporters in phase 3. Phase 3 are all transmembrane transporters. They push that conjugate out of the cell into the cell matrix around that, where it joins into the lymph, then the blood circulates, and then we filter it out. Now, phase 3 is continuing when the liver transports the conjugate from the blood into the liver. That's phase 3. And when the liver dumps it into the bile transporters, that's phase 3 as well. And so as we're coming up the liver pathways, you're going to see that liver transport is synonymous with bile transport. So you see how these things go together. Now, fortunately, all this chemistry is upregulated by NRF2. If this were a cell and this circled the nucleus, there's this stress sensor complex out in the cytoplasm. It's two parts. It's KEAP1 and NRF2. So NRF2 is held in place by KEAP1. It keeps it in place, even though that's not what they meant. It sounds that way. So KEAP1, we see here, is changed in shape and oxidative potential by enzyme inducers. It's actually oxidized and changes shape, and NRF2 gets released and moves into the nucleus and catalyzes the transcription of all the genes for the chemoprotection. These are antioxidants, modulating enzymes, phase 2, phase 3, molecular chaperones, all this for cleaning up the cell. So when the cell's dirty or oxidized or there's chemical toxins in there, that can trigger this. However, this mechanism gets a little sleepy sometimes, and it gets a little blocked by things. And if you're in fight or flight all the time, it doesn't get turned on, because the autonomics are prioritizing which chemistry can get pulled out. And we're going to see there's a lot of this prioritization language. When the autonomics have you in fight or flight, that's going to prioritize fighting or fleeing. And so your energy is going to go to that. All these reactions of detoxification take cellular energy. And so if you got to run away from a lion, you're going to put it over there. Every time I'm trying to run away from my wife, I know that I am failing to detoxify because I'm shuttling all that ATP into my fear. Did I say that? All right. Well, whether it's credit cards or bills or debt or your employees or your bosses, all of this creates this little paranoia situation, which is keeping you from doing that. And chemical stressors can keep you from doing that, even though the chemical stressors are supposed to be activating detoxification. So part of our goal here is to get this mechanism working again. Now, what activates it? Turns out, we used to think it was antioxidants that do it, but it's actually really pro-oxidants that do it. And some of the things we thought were antioxidants, like green tea extract, EGCG, lipoic acid, turns out they're little free radical generators. And just like oxidative stress does this, so do these phytochemically generated free radicals. Now, what you want is, because this is a hormetic stress, you want a free radical generator that doesn't have secondary negative, collateral damage around it. And that's what these plant compounds do, is they generate little microhormetic stresses without having secondary problems. In fact, arsenic, methylmercury, phenobarbital are all NRF2 upregulators, but they got a lot of collateral damage. So they generally group into polyphenols and sulfur compounds. Polyphenols like lipoic acid or pine bark extract. Those are very good at doing that. But even stronger than that are the sulfur compounds. In general, you have sulforaphane and lipoic acid as your major players there. Sulforaphane's a little stronger, but it's also a little bit more cytotoxic, and it's more immunologically reactive, and you make a lot of sulfur compounds from it through CBS upregulation. That's a gene enzyme system called cystabeta-thionine synthase that reacts to very aggressive sulfur chemicals. What's an aggressive sulfur chemical? Bite into a clove of garlic, and that taste is, allicin is an aggressive sulfur chemical. Eat wasabi, and that burn is an aggressive sulfur chemical. The strong smells and tastes in cabbage and broccoli sprouts, those are aggressive sulfur chemicals. And the body, people who are toxic tend to make metabolites of that are not so good for them. So I tend to stick to polyphenols and lipoic acid. So systemic detoxification. Now, if we look at this diagram, you see the cell membrane. Imagine we're in a giant cell up here. This is phase 1, moving down to phase 2. So oxidative activation. Now, a key thing here is when we activate in phase 1, we're making a reactive intermediate. We're making a toxin even more toxic. So it's really important that phase 2 comes in right behind it. And then phase 3 to mobilize things. Now, phase 3 takes it out of the cell to the blood, from the blood to the liver to the bile flow, and bile flow to the GI, or from the blood into the kidneys, kidneys to the urinary stream, and to the toilet. So what happens is phase 2 and phase 3 are linked, and they upregulate and downregulate together. These phytochemicals upregulate them, and inflammation downregulates them. Whereas phase 2 seems to be distinct in its evolutionary pathways, and it has its own triggers, and the presence of the toxins themselves are the triggers for it. So if the toxins are there, but inflammation is downregulating phase 2 and phase 3, you'll generate a lot of reactive intermediates that are not detoxified in 2 and 3. A lot of these are cancerous, these very reactive intermediates. So from a more, naturopathic level, the cell to the blood, blood to liver, GI, or through the kidneys. Now, one thing I want you to see up here, look in this cell. Look at all this folded up space. All that is membranes. The cell is filled with membranes, and membranes are catalyzing the activity, the ability of cells to catalyze reactions that are not favorable in just bulk solution. If I made a glass of these chemicals all together, these reactions wouldn't happen. But the membranes bring things together into a tight zone, so the reactants are right next to each other, and their membrane potential, this electrical potential, drives the ability to bring these things together into these reactions in otherwise unfavorable conditions. The miracle of life is completely catalyzed by the membrane. All right, so if we look into a cell now, and this isn't any cell, this is the hepatocyte, the liver cell. And there's this direction we want. It's this way, from blood to bile. So every liver cell is fed on by blood on one side, drained by bile on the other side. And what are all these gates going in and out with these arrows, big arrows, small arrows? Big arrows represent high flow, small arrows represent low flow. So under good conditions, you have high flow in. OATP moves toxins in. These starbursts with the blue rim are toxins. If they have an X next to them, they've already been conjugated to glutathione, glucuronic acid, or sulfate in phase 2. They're transported in. They move all the way over here to the canalicular membrane. The canalicular membrane is the drainage of the bile tree. So the bile tree, it's like an upside down tree with roots reaching up into the liver, and they go progressively finer into tiny rootlets called the canaliculi. And so every cell is draining bile and toxins. So what's this transporter over here? So this conjugated toxin has come across, and it goes into MRP2, and this little green circle represents bile salts or bile acids. And the toxins and the bile are both transported by MRP2 into the bile canaliculus. Right next to it is BSEP, bile salt export pump. We can guess what that does. It transports bile salts. And these two are inviolably bound. They are upregulated and downregulated together. They are always right next to each other on the membrane. And that's because bile and toxins move together. So when you have cholestasis, the failure to move bile, you have toxostasis, the failure to move toxins. We see a lot of damaged people, very mold damage, a lot of Lyme, a lot of exercise people who work out too far. They and over exercise, they have damage to this area, and this area is flowing very, very slowly. And so we really have to work on increasing that bile flow. Now, this unconjugated toxin comes across, it's come from the lungs or from the GI. It hasn't been processed yet. Goes into phase 1. Notice it turns red because it's more reactive after phase 1. Then phase 2, it gets an X tacked onto it. Then it comes over here. One other transporter to draw your attention to is the MDR, uses phosphatidylcholine to thin the bile flow and keep it moving. And where does it get phosphatidylcholine? It's getting it from the membranes. This is the dominant component of the membranes. So phosphatidylcholine is being synthesized from choline, or it's being brought in raw from membranes in your food, or from when we take phosphatidylcholine either as a liposome or as a supplement on its own. When you run out of phosphatidylcholine, you immediately become cholestatic. All right. So these other transporters, NTCBs, are bringing bile salts into the cell. Where were they getting bile salts? From the blood. Where did that come from? From the GI tract. Unused bile, lower in the GI tract, gets pumped back into the blood and brought back to the liver to reuse it. Costs a lot of money to keep making bile, so you reuse your bile. So there is a banking system of bile salts in the blood. And when the banking system senses too much bile not in the blood, but in the hepatocyte, it dumps it out. Why? Because bile digests fats and it'll digest the cell membrane. So it's got to get rid of it. The sensors also sense when there's too many toxins in the cell. So if this all gets downregulated, toxins and bile salts build up in the cell, and the cell has to do something with them, and it invokes these transporters here, 3 and 4. 3 and 4, they have a small flow here because the net movement is this way. But when we get to an inflamed cell, let's just go right to it. When we get to an inflamed hepatocyte, now there's been strong downregulation of the flow out of the hepatocyte into the bile canalicula's. You have a buildup of all of these things. Notice a lot of unreacted intermediates because phase 2 and 3 are downregulated by the inflammation, but 1 continues. You have a buildup of bile salts dissolving the cell and all these toxins, and there's also free radicals in there. So what does the cell do? Upregulates 3 and 4. 3 moves toxins out into the blood, and 4 moves bile salts into the blood. We downregulated OATP and NTCB, bringing things in, and you can even recruit an MRP2 from this membrane over to that membrane to dump back into the blood. That's the back flush back into the blood. So what kind of things do this? What kind of things block this movement of bile out of the cell into the bile canalicula's? And what is this called? This is called intrahepatic cholestasis. It's different than extrahepatic cholestasis, which is sludge or stones in the bile tree or the gallbladder. This is failure of the bile to be moved out of the cell into the bile tract in the first tract, intrahepatic. So I'm going to go down to this one here because endotoxin is our primary driver of blockage here. What's endotoxin? It's also called lipopolysaccharide. These are pieces and parts of cell membranes of bacteria that have gotten into circulation. Now, how would they get into circulation? Primary way these days is leaky gut. So not the whole bacteria, but parts of it. Why is it an inflammagen? Because your immune cells read the parts of the membrane. Remember when we talked about xenophagy, we take apart the bacteria, we read parts of the membrane, and we put that signal out to the immune system. So here the immune system reads parts of the bacteria that should be in the gut. It thinks you have a septic infection, and it winds up inflammation really hard to kill these bacteria. Where else can you get them? Periodontal disease, periodontitis, a lot of bacteria build up in there. You get endotoxicity from that. In fact, there's good studies around depression and periodontitis. Depression is a manifestation of inflammation in the brain, and endotoxin being the primary inflammagen. You can get them from UTIs. You can get them from jawbone infections, from root canal infections, or NICO, or what are called cavitation infections, chronic sinus infections. All of these things create endotoxemia, which create a systemic inflammation, which downregulate detoxification reactions. They also cause inflammation in the brain called neuroinflammation, shifting you over naturally into a fight or flight sympathetic autonomic dominance. So think about a cellular sympathetic dominance and a neurological sympathetic dominance, all happening at the same time as a result of this endotoxemia. Now, other things that do it are estrogen dominance. And estrogen dominance brings with it, it blocks the liver, but it also upregulates glutamate in the brain. So in the brain, you've got sympathetic and parasympathetic. Sympathetic is associated with the neurotransmitter glutamate. So glutamate over excess, like glutamate excitotoxicity, puts you in sympathetic. And what does estrogen do? It hyperstimulates the glutamate receptors. So for a given amount of glutamate, you have high firing. Now, interestingly, progesterone, a hormone which is incredibly bitter, so it can be considered a bitter, opens up the liver, opens up bile flow, and the metabolites of progesterone are activating the GABA system and making the GABA system more tonified. So there's this inseparability of the neurological system and the liver system. Now, if we go back, this has been known a long time that endotoxin blocks the liver. Back in days of studying non-alcoholic and alcoholic fatty liver, fibrosis, cirrhosis, they knew, this is 1979, they knew that intestinal endotoxin was blocking the detoxification reactions in the liver, but they hadn't even identified what those reactions are Now they can take those reactions, phase 1, phase 2, phase 3, especially phase 2 and phase 3, And they can put endotoxin into the, expose the cells to endotoxin, it downregulates those reactions very strongly. All right, so now we block liver, we block kidneys. We may or may not be dumping out of the cell, but even at a cellular level, we turn down all this stuff from the inflammagens. So we have inflammagens coming in, blocking all this, activating neurological inflammation, and this is a bad situation. Now, if the endotoxin is coming from the GI, what kind of things let endotoxin in? What kind of things cause leaky gut? Now, alcohol breaks down the tight junctions and causes a temporary leaky gut. NSAIDs cause a much longer term leaky gut. I remember having herniation of my disc and really bad sciatica, and I took tons of ibuprofen from it, and that just opened up my GI for a long time. It was really bad. Infection, surgery, food allergies are a big one. It's just every time you eat something you're reactive to, you have all this inflammation in the GI, and it opens everything up. Just carbotoxicity, eating carbs all the time. Roundup, that's a big one for opening up the GI. And the carbotoxicity, we'll see when we find AMPK activation, that does seal up leaky gut. Bonus content number 1. I didn't expect that slide. Thank you, Justin. I feel like I should be on a game show. And for bonus content number 1, Justin, answer this question. All right. So bonus content obviously is talking about products, right, Justin?
That's right. That's right. We want to know what to do now.
What to do. And so the whole talk's set up that way. Here's your pure education. Here's your application. So we'll do this. We'll do the application on detoxification. That'll lead into showing that some of this is AMPK. Then we'll do pure education around AMPK and so on and so forth.
Correct. All
right. So this detox system is the core of all of our detox protocols. And this is a 2-part system called push-catch. The push is this stuff called liver sauce. Don't you like that? It's like A1 sauce on your liver. And that activates all this movement, the push from the cells to the blood, the pull through the liver, the dumping of the bile. It's activating that whole drainage system into the GI tract. But one of the problems is a lot of these things reabsorb. So the catch is a system of binders. There's several different binders with different chemistry. These are things that go through the GI tract, like clays and charcoal, that bind all those toxins and take them all the way out. So push catch is the core. Then you may have different add-ons, whether you're metal toxic, mold toxic, you have some viral underpinnings. There are different ways to modify that. So the push, we'll talk about the different programs that are in the liver sauce, but bitters, silymarin, phosphatidylcholine for bile flow. This is why traditional bitters were a big bile flow thing. This is why it kind of fixed everything. Turn of the century, you took bitters for whatever ails you because it moves your liver, and that just makes you feel good. There's an inflammation control and NRF2 upregulation. A lot of these toxins wind up mast cell activity, immunotoxicity. The immune system reacts. How does it react? It releases pro-inflammatory compounds, and the pro-inflammatory compounds that are pushing down detoxification. So when we move these toxins, we need control around that system, and then the broad spectrum binder to catch everything. So looking into liver sauce, I like to talk about programs in the product. So programs are groupings of ingredients. So I kind of, prepped you for like I'm some monodimensional guy that puts one compound in a nanoparticle, but it's actually not true. And so here are these groupings. And this top one, these are whole plant extracts of myrrh, gentian, dandelion, and solidago. In fact, we have a product called Bitter X that is these 4 products in here. Phosphatidylcholine is present in all of our delivery systems. And so this is primarily responsible for the bile flow. There is help from silymarin, which anchors the transporters and stabilizes the transporters. Silymarin kind of does a lot of different things for us. So that's the first program, is the bile flow. And then the immunotolerance and mast cell stabilization. So when the immune system starts when you get all toxic, you have that Th2 shift. You start being allergic to everything. Your mast cells activate, release pro-inflammatory stuff. So every time you eat something, including some of the products you're taking to try to detoxify, you may react like that. So we have mast cell stabilizers, quercetin, luteolin, and DIM. DIM, people know DIM as a crucifer extract that's helpful for estrogen metabolism, but it turns out that it's really good for immunostabilization as well. Whereas luteolin and quercetin work on stabilizing mast cells, DIM is working on the other immune cells towards TH regulatory dominance. TH regulatory dominance is leading the body not to be so damn reactive to everything. And then DIM, lipoic acid, and silymarin together are NRF2 upregulators, that big trigger to turn on all of your chemoprotective system. All right, so there's Bitter X. That was the first 4 ingredients. And that's working on this bile transport, both on a gallbladder level, but also on a hepatocyte level. Phosphatidylcholine is working on the MDR to help fluidize the bile and keep moving that. You'll see a lot of people get sludgy bile. And so sludgy bile is just not moving bile fast enough. And so this is to stimulate all that movement. In fact, I'm always asked, if I had my gallbladder removed, can I do this detox system? Well, definitely you can. So think about the whole movement of bile. So at a cellular level, you're producing bile and secreting it out of every hepatocyte into the canaliculus. They're coming down into larger vessels and finally coming into the common bile duct. And then the bile is accumulated in the gallbladder, that sac. And then it's released in bolus doses when you eat. Eating stimulates the vagus nerve and squeezes out some bile so that the bile can have this aspect of a digestion as being a fat emulsifier. But the cells never stop generating all that bile. And so what happens is people who lose their gallbladder usually have kind of sluggish bile. They may have poor diets that don't lead to secretion of a lot of bile. And so everything kind of locks up and freezes in there. So you cut off their gallbladder because it's filled with junk. Now, really, if you're taking a lot of bitters and PC for a long time, you'll clear a lot of that stuff out. You'll be able to keep it. But the fact that you lost the gallbladder means you were kind of sluggish in your bile production. And you could do this whole detox system, but you should probably go a little slower because you secrete slower. Now, the other thing that PC does is feed all of our membranes. Remember, I've been talking about membranes kind of romantically, is that they're like battery, they're like generating power, like the biological power of the cell is dependent on polarization of the membrane. If you're into Bruce Lipton and his ideas on epigenetics, that's all powered by the membrane potential and the ability of it to set a potential. Now, the outer membrane is also communicating through the extracellular matrix. All of these fibers of the extracellular matrix are where all the transmission patterns are going through. This is where all the communication is happening. This is where the meridians of TCM are. This is probably where that light-based communication that enables you to muscle test is going through. In fact, Bill Tiller from Stanford describes these hyper-ordered ion channels that go through there that he says are the meridians. But as you go deeper into the cell, I don't have the pictures, but remember the picture inside the cell of all the membranes. Now, transmission of communication is coming inside to the intracellular environment, and the membranes of the mitochondria, the endoplasmic reticulum, the Golgi apparatus are all communicating with each other and feeding information into the nucleus to decide which of the programs in the nucleus can be manifest at that time based on the resources in the extracellular matrix that are available to the cell and the biochemical power that is available to the cell in the inner intracellular space. So that's all membrane-based communication. So the more powerful your membranes and the more often you recycle those through autophagy and make better membranes with more power, the more biological power you have in the cell. That's it's very stimulating, isn't it? I want to have biological power in my cell. All right, so immunotolerance and mast cell stabilization, just to say very quickly, quercetin and luteolin, very poorly absorbed, but very well known as mast cell stabilizers. They're used for that. DIM, shifting you over to T regulatory dominance. Also DIM, able to reverse epigenetic blocks to NRF2. And this happens a lot in mold toxicity. And then NRF2 upregulators, remember that master switch that brings everything up, that's DIM, lipoic acid, and silymarin. So we get all that engaged. We get the bile flow. We stabilize the immune system. We'll upregulate NRF2. We start dumping all these toxins into the GI tract. And what happens? they get reabsorbed back in your blood through enterohepatic circulation. So enterohepatic recirculation is made worse when you have leaky gut, but a lot of toxins like methylmercury from fish, 95% reuptake. So you need, when they get down there, to have a binder to get them. And Ultra Binder is our multi-specific toxin binder. So each binder has its specificities. A toxin that a small amount of binder gets a lot of toxin. And then things it's not specific for, you need a shovelful of binder to get a little bit of toxin. So you want a bunch of different toxin binders. IMD is our proprietary thiofunctionalized silica, which has very high specificity for heavy metals. Then we have charcoal and zeolite, each with a range of different specificities in the pesticides, the herbicides, and very importantly, mold toxins. And then chitosan, which is very good. It's a molecular mimic for Welchol, used in the Shoemaker biotoxin protocols, including mold toxins. So all these work together to get the whole soup of toxins, then acacia gum and aloe for rebuilding the lining of the GI. So now when these toxins dump into the GI tract, the binders come in and pick them up and take them out the fecal excretion. Now, getting all these mechanisms working and upregulated, downregulated might take a little time to entrain your body into being able to toggle back and forth between these states. So we titrate up. And in the protocols, we have like a level 1, a level 2, and they follow this idea over time of increasing dosage, getting to an intensive where you're really just dumping things out and reinforcing the power of this system, accessing genes that haven't been turned on for a long time, getting the body used to engaging this deeply. And this will carry through over time. Once you've brought these genes out, you stuff them, it's like a dirty old office. You stuff information down under piles of paper and you access them again. And, oh, I remember all this stuff. And the body is engaged back into doing this, even when you're not actively on the detox. And then after that intensive, you can come down to lower levels. And we lead you through this in the instruction booklets. And what's the power of this? There is a study done on fatty liver where Texas Center for Lifestyle Medicine, Dr. Cheng Yuan, ran 102 people through doing this protocol, and he just led them on a twice-a-day dosage straight across. In 1 to 2 months, he resolved 82% of the fatty liver cases, 72% of the biliary sludge, and in the gallstones, instead of taking the gallbladder out, he reduced, he had 3 of 12 gallstone patients had complete elimination. The other 9 had improvements in size, with the biggest one being 1.73 centimeters. That's pretty big, reducing down about 30-40% to 1.14 centimeters in 32 days, 1 to 2 months to create these dramatic changes. So why do we get such dramatic changes on fatty deposition in the liver? Now it's easy, it's enticing for us to say, well, you move the toxins out and everything works. Part of that's true. But it turns out half of these ingredients are strong AMPK activators. We activate AMPK, it brings with it autophagy, which brings with it lipophagy, which is burning away fatty things. And it brings, that's all through something called mTOR inhibition. And so this is why this feeds into the whole keto thing, is when you're high on carbs, you're mTOR forward, you're laying down mass. When you're low on carbs and also branched chain amino acids, so low on carbs, low on protein, you mTOR block, and that makes you go inward and burn up all your resources. Not in a bad way. You're also recycling. So all these bad cells, you're burning them up and creating new cells. And you're also changing levels of NAD in the cell. So in the next section, we'll go into what all that means. So now let's talk about, back to education, AMPK and the applications to metabolism and detoxification. So AMPK is the central switch that's toggling back and forth between fed and fasted. And here, take fat out of the equation because you're going to have fat on both sides. Fat can be a fuel even when you're having fasted activities. And that's why the keto diet can be so effective. So in fasting, what happens is there's an AMPK activation. Now, this is generated by burning up all your ATP and resulting in AMP. So AMP is sensed by the AMP kinase and activates AMPK processes. Now, AMPK can be brought up a little or brought up a lot. And if it's activated through some of these channels, one called LKB1, liver kinase B1, this is where nutraceuticals and pharmaceuticals like metformin come in. Now you can activate it a whole lot. And you can activate it even if you still have carbs on board. But it's better if you take this stuff while you're in a fasted state. And that is simple. I mean, just intermittent fast in the morning, no food, take this stuff and you get a hyperstimulation of the system. And that results in blocking of this thing called mTOR, which then blocks protein synthesis. And it activates this ULK1, which activates autophagy. It activates adipose triglyceride lipase, which activates fatty acid catabolism. And it blocks acyl-CoA carboxylase, I think it was, which is then in turn blocking fatty acid synthesis. So you stop laying down fat and you start burning fat. So it's a really important mechanism. And really, it's, this is the Tao. It's the pendulum. Fed, fasted, fed, fasted. I got to lay on more mass. I do fed. I put stuff down. And then I want to clean up. I go into fasted. I clean up the excess. And now, a lot of this, notice the trigger was liver kinase B1. The signals for this are coming out of the liver, and they're very active in the liver. And this AMPK is a target for working on things like hepatic fibrosis. So here in this graphic, we have things driving us into fibrosis, like viral infections, hepatitis C, alcoholic and non-alcoholic fatty liver disease, biliary obstruction. Carbon tetrachloride is a toxin. So toxins, infection, and fatty buildup or physical obstructions all leading to fibrosis. And really, I like to look at it as healthy, fatty, fibrotic. So fatty is the first step down to fibrotic and cirrhosis. So once fatty happens, you're on your way because now you have a constant generation of inflammatory cytokines in the liver. And the inflammatory cytokines are what generate the fibrosis. And so that's really your steps. And you want to reverse those. And you want to go back through those levels. You can take adiponectin is one of the hormones that's released, especially when you fast a lot. And when you have a healthy low level of fat, you have a high level of adiponectin, which is driving AMPK activation. Nutraceuticals, curcumin, anthocyanins are driving it that way. Pharmaceuticals like metformin and fenofibrate and whatever that one down there is, all driving that backward that way, all through AMPK activation. Now, AMPK activation, here we have the AMPK being activated or phosphorylated by liver kinase B1. Then when we look at like activation of the mitochondrial genes, you have to phosphorylate them and then you have to deacetylate them. So we'll see you need AMPK, then sirtuins to get the most out of all this. And when that happens, we block all this formation of new fat. We turn up catabolism. We burn down the fat. But look, remember we were talking about membranes being the key to this. AMPK activation repolarizes membranes and puts their power back in trace. It also changes cytoskeletal organization. So you're going to get rid of extra fiber and you're going to tighten up tight junction formation so you don't have leaky liver. Leaky liver, who'd have thunk it? We thought it was just leaky gut. Turns out there's leaky gut, there's leaky liver, there's leaky blood-brain barrier. And my friend, we won't even talk about that, but there's other parts that get leaky too. She talks about women's health. And very important, we upregulate canalicular trafficking. I'm sorry, Justin, I was going to come out and lecture for you out in Oakland, but I was stopped at the airport for canalicular trafficking. Son, you're moving bile too fast. And that's what that is. Canalicular trafficking is that movement up at the canalicular membrane. And do we have evidence that natural products can work on this? Yes, we do. I just showed you great evidence of AMPK upregulation working on fatty liver. But here we have AMPK as a potential target for fatty liver. Here we have quercetin working on it. Here we have silymarin working on it. Well, we put all that stuff together and we make it happen really quick. But all these papers talk about why do you get fatty liver? Because you're not going into autophagy. You're not breaking things down. And one of them goes so far as to say it's all from long-term nutrient oversupply, eating too damn much, and it's really around carbs. But remember, proteins can do this too, because the mTOR forward switch, insulin drives it harder than anything else, but branched chain amino acids drive it too. So eating too much protein drives it. That's where paleo, with its overemphasis on protein, even though it carb restricted, never got the results that keto did. And here we have AMPK activation leading to increased formation of tight junctions. We're going to skip through this quick, but it also decreases hyperactivity of the immune system to the foods that come in. So lowering reactivity and tightening the tight junctions. On a cardiovascular level, AMPK blocks NF-kappa beta signaling. NF-kappa beta signaling, when you block it, you lead to more polarization, decreased cytokine release, decreased lipid accumulation, decreased leukocyte homing. That's how plaques are built. That all reducing vascular inflammation and then improving vascular function. You have all these things that are upregulated here. And here are some of those triggers that we were talking about before. Sirtuins, PGC1A, FOXO. These are all the things that lead to longevity, that lead to free movement of blood throughout the body, opening up the chi flow. And all this decreasing inflammation. Where does the inflammation come from? It comes from fat. So AMPK. Here, the interesting thing to catch is diet-induced obesity is associated with systemic inflammation, which is considered to originate predominantly from the adipose tissue. They're calling quercetin and resveratrol anti-inflammatory and anti-insulin resistance. The inflammatory part, they're only anti-inflammatory. It's not like you hit your elbow, oh, I'm going to take some quercetin, it'll make that hurt less. No, they're decreasing lipid accumulations, and that's decreasing the release of the pro-inflammatory cytokines. All right, so we're actually going to we're going to go right into talking about NAD, and then we'll talk about the products for that at the end after that. So NAD is the everything molecule. So NAD is involved in redox reactions. Part of it is during oxidative reactions and taking electrons away from your carbohydrates and your lipids and bringing them into the electron transport chain and driving ATP production. While on the flip side, NADPH is bringing hydride to your oxidized glutathione or re-reducing glutathione. So part of NAD is doing oxidative metabolism, parts doing reductive metabolism. Circadian rhythm. NAD controls all sirtuins, and sirtuins control the clock genes, which control circadian rhythm. When I started taking NAD, I finally started getting deep sleep, and I needed to sleep less. It's controlling inflammation because it's controlling the fatty accumulation and that release of NF-kappa beta. It is totally activating DNA repair. When your cell phone is vibrating your brain and vibrating your DNA, it's NAD to the rescue that fixes that. In fact, I noticed way less EMF stress since I started taking NAD and AMPK activators together. Cell division, signaling, epigenetics, it is responsible for cleaning your genome, turning on the stuff you want, turning off the stuff you don't want. Keeping top of mind the most premium of the gene sets. Realize, depending upon your energy and depending upon your NAD levels, your body chooses the premium gene sets or the piss poor gene sets. Just like I said in immunological reactions, I'll either kill the bacteria or I'll negotiate it with it. And so this is all bioenergetics, and NAD is driving bioenergetics, and it's doing it in the brain, the liver, vasculature, heart, hepatocyte, everywhere in your body. NAD is driving energy. Conversely, lower NAD is leading to obesity, fatty deposits in the liver, problems in the heart, all these different pathologies. So when it's high, there's vitality. When it's low, there's pathology. And of course, like all good things, we lose them with age. So here we have NAD in brain tissue going down with age, NAD in skin going down with age. You bring these things up, you get your vitality back. This was David Sinclair's information theory of aging. So information, I've been saying this again and again. All the programs for all the ways you could be, to be a baby, to be an old man, to be shrivelly when you're 50 or to be healthy when you're 50. Those options are all within your nucleus. They're all within your DNA. And you have to have the biochemical opportunity to manifest them. You have to have the biochemical resources, the biochemical power to manifest the higher levels of your potentiality. And it's NAD, which is determining whether those go on or off. And thus he has the information theory of aging. Now, when it gets into the mechanisms of sirtuins, sirtuins and FOXOs are these premium genes, these premium pathways. So we used to talk about things like resveratrol as sirtuin activators, and resveratrol gets the sirtuins up. But sirtuins are activated primarily by NAD and secondarily hyperactivated by the sirtuin activators. Now, here we have a sirtuin going in, and they're called deacetylases. There's if there's a gene that we want to activate it, or a protein we want to activate, we have to phosphorylate it and give it like this active potential with the brightness of the phosphorus, and that's from AMPK. And then we have to take the silencer off of it. It's always got a silencer, and that's called an acetyl group. So the sirtuin takes the silencer off. And so here we have the sirtuin deacetylating it, the NAD activating a sirtuin to deacetylate the target. Here we have a stacked sirtuin activating compound like resveratrol, like quercetin, EGCG, curcumin are also ones. And now that's hyperactivating it, making it able to work on a bunch of acetylated targets all at once. But what blocks that is nicotinamide. Nicotinamide is the spent product of NAD activating a sirtuin. So when NAD is done activating a sirtuin, it's nicotinamide, and it sits on that site there, and we have to get rid of it. And that's the key to making NAD work, is having a lot of NAD, but making sure we don't have excess NAM. And when we talk about the integration of things, we'll talk about the NAD cycle and the methylation cycle. So now for the bonus content, protocols and products. So some of the phytochemicals that are great for AMPK activation. Resveratrol, that's long been known, kind of a fasting mimetic, exercise mimetic. Berbeриne's long been known as nature's metformin. It's really good for blood sugar control through controlling insulin resistance. Quercetin is very good. It activates mitochondrial biogenesis. It's a sirtuin activator. It's AMPK activator. Milk thistle, we saw milk thistle working on reducing fatty liver through activating sirtuins and AMPK in the liver. Dianylyl methane, or DIM, we knew it for hormones. Then we knew it for immunotolerance. Now we know it as an AMPK activator as well. And cinnamon has long been known for that. So all those are stacked together in these nanoparticles of AMPK charge. So how do we know that AMPK charge works on all these things? We know theoretically it does, but if you take this in the morning before you eat, night before, doesn't matter what you did. You drank beer, you ate French fries, you ate pizza. In the morning, an hour after you take this, you will be in ketosis, dietary ketosis. You can measure your blood ketones. They'll be over 0.5 millimolar ketones in the blood. And that signaled that you just strongly struck the AMPK bell, and all the cascade of the metabolic signals after that were putting you into burning all of your resources that are stored in there. It's a beautiful, beautiful long-term metabolic fitness and anti-aging aspect, because a lot of these are sirtuin activators as well. And just to overlap the AMPK charge and the liver sauce, these can be done in different ways. Remember the push catch. So we're going to have liver sauce activating detoxification, then ultra binder binding it. Well, most people have heard of the keto flu. And the keto flu is release of fat-soluble toxins. I mean, there's aspects of electrolytes and water balance, but it's fat-soluble toxins more than anything. As you burn the fatty deposits, the toxins come out. Now, a lot of these things that are in AMPK charge are also in liver sauce, quercetin, milk thistle, DIM. And then the bitter that's in it is berberine. So we're activating a lot of liver pathways with AMPK charge. And sometimes in the first couple of days of taking it, people will feel detox reactions. So we will often then couple it with the ultra binder and have a push catch that's more metabolically based. We have one that's more toxin based, liver sauce and binder, one that's more metabolically based, AMPK charge and binder. One of my favorite things to do is have people do a month or 2 months of liver sauce and then another month or 2 months of AMPK charge, even 1 month of push catch liver sauce binder, 1 month of AMPK charge binder, and then keep going on the AMPK charge. But you've done a lot of detoxification at the same time that you're building up all of your metabolic fitness. Now let's get into NAD. All right, it's coming in the next one. So NAD and methylation are inviolably linked. Well, NAD is the highest end of the B3 cycle. So niacin and niacinamide can become NAD. Now we're using the highest levels of NAD precursors, nicotinamide mononucleotide. There's one other really good one called nicotinamide riboside, but we use nicotinamide mononucleotide, the immediate precursor to NAD. We put it in a nanoliposome, and we do that so we can get high absorption of it into the blood, get it into the cells where it can become NAD very quickly. Then as NAD, it can activate the sirtuins, but then what happens is it becomes nicotinamide, and we have to get rid of it because nicotinamide is blocking the sirtuins. How do we get rid of it? We methylate it with SAMe. So we need support for the methionine cycle and the cycle that feeds that, the folate cycle. So we need methyl support. So we pin together NAD gold, the liposomal nicotinamide mononucleotide, with methyl charge, the cofactors for generating SAMe. And just to see how that all works, so nicotinamide mononucleotide becoming NAD and activating a sirtuin. We got to put that one in sooner. We got to change that cycle there. When it activates the sirtuin, it becomes nicotinamide. Now, nicotinamide normally regenerates itself back up to nicotinamide mononucleotide through the agency of this enzyme NAMPT. But NAMPT is strongly held back. It's strongly repressed with age and any inflammatory activity. So the nicotinamide is not becoming more nicotinamide mononucleotide. Hence why we supplement in with NMN. So now in doing that, we're building up NAM, but we have to get rid of it. And we do that by peeing it off after methylating it with SAMe. So we're pooling in with, we're bringing in NAD with NAM, with NMN, building up NAM, then methylating it to get rid of it. So we need the methylation combined with the NAD formation. When we do all that, you're just driving cellular energy all the time. Now, we also kind of want a sirtuin activator with it. So we need NAD formation, we need methylation, and we need sirtuin activation. So you've got NAD, gold, methyl charge, and then for your sirtuin activation and AMPK activation, we use either AMPK charge or the one. Now, what's the difference between the two? AMPK charge is more metabolic, trying to take off fatty deposits, trying to have more autophagy, maybe, burning down, some of the wrinkles and regenerating stuff. Whereas the one is more straight mitochondrial biogenesis. There's more neurological energy. There's a little bit more, there's more phosphatidylcholine and adaptogens in there. So a little bit more adrenal and mitochondrial build. Choose one of these two, or as some of my friends do, and I do, I just rotate back and forth between the two and take the NAD gold and the methyl charge NAD. I guarantee, and the methyl charge, I take those every day. I guarantee this will be a game changer in your energy. And it's one that, yeah, you'll get more energy right away, but you're going to see yourself change. You take this stuff for a year, you're going to see and feel dramatic changes in your metabolism and your body. Now, we have one all-in-one solution here called NAD Platinum. We put all the ingredients from NAD Gold and methyl charge in there, so you have those balanced. And then we added some sirtuin activator, resveratrol and quercetin, and the senolytic quercetin. Senolytic, so when cells get old, they don't get to the Hayflick limit and die, but they're too old to do anything anymore. They go into senescence and they start releasing pro-inflammatory cytokines that also recruit other cells into senescence. So they call them zombie cells. So you want a senolytic for anti-aging support, and quercetin is a great senolytic. All this is based on bioavailability, how you get things into the body. Just to say it really quickly, we use nanoliposomes and nanoemulsions. Nano means it's under 100 nanometers, and that's where things absorb into the blood super, super fast. If it's a water soluble, it's a liposome, a water droplet inside a shell of phosphatidylcholine. If it's a nanoemulsion for a fat soluble compound, it's got a little lipid drop with all your compounds in there in a shell of phosphatidylcholine. And this is uptake in a cell versus size. The smaller the liposome, the faster everything absorbs into the cell. And when you get this, when you make these products, we're the only company that has control over the products. We use dynamic light scattering and electron microscopy to characterize the sizes, to decide what is the most stable size and what has the highest uptake. And then we pin that size range into the specifications of the product, and we measure with lasers the products as they're coming off the manufacturing line to ensure that every batch is the same size and thus the same activity. And just so you know, when you go into liposomes and nanoemulsions, when you're in the nano world, they're optically transparent. Their particles are actually smaller than wavelengths of light. So if you see a bunch of products like these, whether they're liposomes or emulsion, you see they're cloudy, you see they have this chunky stuff on the side. These are all over 300 nanometers. You're not going to get that fast uptake. You'll get a little bit more uptake, but not what we get. CBD, 6 times uptake over native CBD. Quercetin, 25-fold increase in bioavailability. Curcumin, 300-fold increase in bioavailability. All this kind of stuff comes in not just higher level, but immediate, peaking between 15 and 30 minutes in the blood, activating all these processes. That's why in push catch, you can take your liver sauce, move all these toxins down into your GI, 30 minutes later, take your binder, catch everything that's there. All these things leave the blood. You stop detoxifying. You have this nice little loop of detoxification in a 30-minute window. Give it 30 minutes to clear out, and then you can go about eating, or you can keep fasting, whatever it is. Only when these nanoemulsions do you get those kind of results. And here's the CBD. This is native CBD in hemp seed oil with its uptake peaking at about 2 hours versus hours peaking in 25 minutes. We have data showing that some people peak as soon as 15 minutes, 6-fold increase in general bioavailability. And this is the thing too. In the GI, you have over 100% variability person to person of what the blood level is versus the dosage versus the nanoparticle. You're looking at 20 to 30% variation person to person. So now we can predict blood levels. We can have reproducibility. We can have each person going through relatively the same thing that the other one is going through versus GI. You just don't know what's going to happen. This is a comparison of our liposomes. This is a CACO2. This is uptake versus these different products. This is a control here. CACO2 is a GI membrane model, and this was 6 different liposomes of glutathione from the market. All of them no better control or significantly worse, and all of them with significantly degraded glutathione. So it's not just, oh, Chris told me about liposomes, this one's cheaper. Good luck with that. 30% a label claim, 42% a label claim. All these products are degraded and don't absorb. So it's not just because it's a car that, yeah, it's a Porsche, it might be a Yugo, it might not even start, it might be 20 years old. We don't know. We don't know. That sounds like Trump. I shouldn't have said that. And this is CACO2 uptake for the NAD Gold. We had a 4.25 fold increase in absorption from the liposome over native NMN. So this is the promise of what these deliveries give to you. They give you rapid delivery. They give you unmetabolized delivery, which is stronger. Through the GI, you already start detoxifying it as it comes in, and they just don't work on these biochemical mechanisms like the raw compound coming in these nanoparticles, coming in fast and hard, being able to trigger these processes and then clearing out of the body. So you're not sitting on these compounds all day. So That's what we have. We have detoxification of glutathione leading to immunity and longevity, AMPK activation leading to cardiometabolic fitness, immunity and longevity, and NAD leading to strong mitochondrial health, which gives you strong immunity, strong longevity, strong vitality.
Thank you. So we have some questions for you, and I know that we're a little over time, so I appreciate everyone's with us. And Dr. Shade, I don't know if we can see how much time for each of these questions. We could do it like a game show with like speed rounds as we get to some of these. So it's a great opportunity to have you and have you live with our practitioner community. So here's a couple, just starting from the beginning here. Bartonella infection. So this is, you know, I used to get it from cats, right?
Cat scratch fever.
So this is a patient with the Bartonella infection, tender lymph nodes, both armpits, groin. It's from a dog bite on the thumb, which is red. Do you have any suggestions?
Oh, that one's out there. Micromanager as the antimicrobial in that. And I would do push catch with it. And so take a liver sauce, and depends how cute it is. I like a big dosing of micromanager, so I'd take 3 of the soft gels with a teaspoon of liver sauce, and wait for the binder 45 minutes to an hour, because the soft gels are the micromanager, and the drivers of the antimicrobial activity there are artemisinin, berberine, propolis, and andrographis. Plus, there's some inflammation control in curcumin, boswellia, CBD, and some silymarin and quercetin there. So that's pretty broad spectrum. So do that twice a day, and you should see some results pretty quick.
Oh, fantastic. Micromanager and the push catch. How does altering inductible NRF2 by means of our nutrients, nutraceuticals interact with the constitutively elevated NRF2 in some cancers? Is there any relationship between those two?
Yeah, I mean, that's something that people worry about, is if, so in cancers, there's constitutively upregulated NRF2. So cancers are trying to run antioxidant protocols all the time. 1, because a more reduced cell can proliferate faster, but 2, because they have some breaks in their free radical defense chain. And so they just try to really mop stuff up the best that they can. So people worry about NRF2 upregulators. Yet, what kind of NRF2 upregulators are we using here? Quercetin, luteolin, DIM, silymarin. These are all things that are used as anti-tumor compounds. People worry about using glutathione and glutathione under those cases. And with people with active cancers, I say, no, don't use glutathione. But I'm not even sure there, because it's increasing Th1 activity, which is increasing immune surveillance. And there's some data saying that glutathione helps. But I don't see any problem in the nutraceutical blend, because the nutraceutical blends have been used, to kill cancer stem cells. They have all this data around them as anti-cancer.
That's great. Okay, thank you. A question about cells and their experience of time. They probably experience time very differently than we experience time. How rapidly can cells toggle back and forth between mTOR and autophagy? Is that something that can happen within seconds, or as we experience it?
mTOR forward, mTOR backward. It happens really quickly. I mean, it can happen, if you're exercising enough, and you use up all your excess ATP, you'll pop over into mTOR blocking. With the AMPK charge, formerly keto before 6, you can take it in the morning, and I don't think you're mTOR blocked. Yeah, you're fasted overnight, but in an hour, hour and a half, you start generating blood ketones. So obviously it had switched pretty quick. You can jump back and forth pretty quick. Then you get into lunch. If you have just a little bit of carb in there, it doesn't stop things. If you have a lot of carb, it'll stop you. And so, within the space of 10 to 30 minutes, we see it toggling back and forth. How fast does it happen on a cellular level? Probably pretty quick.
Mm-hmm. I've been taking liposomal glutathione, and it's caused a bit of a stomach upset, but it took the pain away immediately, or amazingly, sorry. But then when it wore off, the pain came back. I seem to feel even more pain, but should I stick with the glutathione, or is it normal to wear off quickly for some people, or is it better to wait for a whole week for the program to play out? And this person also knows they have gallstones.
Okay, if it's doing something acute for you, a lot of these things go in and metabolized out pretty quickly. You know, it's like drugs last a lot longer because they engineer them not to be able to be detoxified, which is a bad thing. So, you know, I'd, you know, be happy with the natural. If it goes away, don't be afraid to do a smaller dose, you know, several times a day, 5 times a day. It's no problem, especially when it's doing something good for you. Just make sure to balance it. Once you start taking a lot, you're going to upregulate detoxification. Make sure you have binders on board. You know, I like push catch, you know, to integrate those two, but get yourself a bottle of binders separately. And anytime, you know, you've been taking a lot of glutathione, you're feeling good, and then all of a sudden you're like, whoa, things are a little funky. Take some binder, just binder on its own. And it is the balm of Gilead. It kind of soothes everything. And anytime you're going through that, take that. Now, you're going to get out of those woods where it's so on again, off again. It'll get more stability as you get deeper into the system.
These are going to be all over the place, Dr. Shade. So, I've been doing the push catch now for 8 weeks, and I assume that they've been doing 5 days on, 2 days off. This practitioners having 1 bowel movement a day at this point. They still need extra help moving the bowels. You know, why are they having this problem? And they're also taking glutathione in the 1. Any suggestions?
Yeah, some people. So with the binders, you know, charcoal and clay, especially, they can be constipating. The IMD isn't. In fact, that helps people. And then you've got some aloe and some acacia gum to help fluidize things. But, you know, I'm choosing a middle of the road path here. Some people are really loose, some people are really tight. And so people who tend to tightness get tighter with the binder. And so all you got to do is take some magnesium with the binder. You can either take some mag citrate, but if you want the more laxative magnesium, you want a magnesium oxygen. And you'll see these like oxymag and mag peroxide and super oxomag. And, you know, it's funny because magnesium oxide used to be like the turkey, you know, because it has no bioavailability, but it has high osmotic capacity, so it makes you poop more. So you just need something to move the bowels to counteract the constipating action. You know, if it's 1 good bowel a day, you may just be fine. But that's how you modulate the effect of the binder on your stools.
Okay, great. Would you switch to Bitter X after you've done the push catch to continue the detox maintenance?
Yeah, well, you know, choose your bitter. And bitters is one of the great maintenance things there because it's working on so many different levels. Bitter X and Bitters 9 have slight differences. Bitter X is more of a liver purgative, more of a bile mover, and has some, you know, digestive help. But Bitters 9 is more warming herbs. You've got some clove in there, you've got some juniper, and it's more of a stomach ache. And so if you're having sort of a weak digestion and not, you know, you just don't feel hungry, you do the Bitters 9. You know, of course, I keep them both on hand and sort of go back and forth between them. And it's not like Bitters 9 doesn't move bile, but it's a little bit more warming and better for the stomach. And Bitter X, more of a liver purgative and more of a uterine purgative for women who have hard cycles. In fact, Bitter X for women who don't cycle well is really, really good. That and CBD.
Great. If I have a patient that I feel that they're pretty toxic, should I start super slow? You know, I think that they probably have a mold toxicity and, you know, and who knows what else.
Yeah. So you can either go to small doses on the liver sauce. Usually when they're sensitive and very toxic, you want to keep the binder high and the catalyzers a little lower. And so you can go to lower doses of the liver sauce. You can buy them independently and maybe do a quarter or a half teaspoon liver sauce and a full teaspoon binder. Or you can decouple liver sauce. You can buy it as independent parts. And so Bitter X and PC with binder is a great push catch.
Mhm.
And you might add glutathione or liposomal vitamin C. The more sensitive they are, the more like C is a nice calming one. It really takes the free radical stuff down without pushing detox too much. So those are, like the simplest and most gentle is C, bitters, PC as you push, and then binders your catch. And also for mold toxicity, binder more often during the day without necessarily hitting the push stuff during the day. So those are ways to break it up.
Okay. I have so many questions on the push catch, by the way. I'm just gonna
Great.
I'm gonna bring them together. Can we do push catch with a patient that has a history of heart disease, or they're in a severe state of inflammation with autoimmune going on?
Yeah, now here's another one, more the autoimmune one, is to start, just like we said before, like, maybe bitters and PC as your push. Now, what do you want to add on there? Say you're trying to control some of that autoimmunity. Maybe you want CBD and DIM with your bitters and, with your bitters and PC as your push side, and then your catch side. That way, they're not reacting to things, and get them a little stability and then move to the core push catch. Heart disease, I'm not worried about there. I might add on PC. So there's not a ton of PC in liver sauce. I often want more. And so I'll look for my add-ons to have PC. So add-ons that have a lot of PC, pure PC, of course, has a lot of PC. Glutathione complex has a good amount of PC. The one has PC. That's a great one for heart because you have CoQ10, you have resveratrol, you have PC in there, and adaptogens. Artemisinin, if you're going after like some GI dysbiosis, and EDTA. So EDTA has a lot of PC. That's also a good heart one. Usually you want to add glutathione in with that too, maybe slowly come up with that. So you got to decide, how you're going to get extra PC in there for the heart, according to what their, the other complicating factors in their life are.
Fantastic. People are loving that you brought up EMF protection, and that the product that you brought up with that was NAD Gold Plus
and AMPK charge. Both of those are very strong on NAD. So the calcium, voltage gated calcium channels, according to Martin Pall, are the biggest target of EMF, and they get dysregulated, making calcium flood into the cell. Well, calcium's all supposed to be outside the cell, magnesium inside the cell. When calcium levels go up inside the cell, it catalyzes these runaway inflammatory reactions. And so the way to fix what fixes that channel is actually NAD. So you need more NAD in there. You can have NAD Gold. The other, more systemic way is the AMPK charge. When I first made AMPK charge, I noticed it was stopping me from being EMF reactive when I traveled. I'm on a plane and I'm like, I got an EMF point I test, it's not there. The sort of head reactions aren't there. And then eventually sort of more direct is the NAD. So those are the two players for EMF sensitivity. I'll tell you right now, like AMPK charge is a long range winner. Like that and the NAD, like if you just give me those two, I'm good for the rest of my life.
Good. Now, do you have to be on a keto diet to use AMPK charge?
Absolutely not. I take it before I eat carbs, and it'll make you burn the carbs more, store them less. It'll have less of that carbotoxicity, that glycotoxicity, they often call it. My life, I intermittent fast. I don't eat it in the morning. Lunch, like today, I ate a keto lunch, and I'm taking AMPK charge, in the morning, and I take a little bit more before lunch. But, so I ate salad with some salmon. But if I had eaten a sandwich, which I do sometimes, I feel like I want some carbs that day, I will still take it. So I burn more. So you do not need to be on the keto diet, but if you're trying to get those effects, then couple the two. But the reason we called it keto before 6 p.m. Like you could be keto all day and go eat carbs at night. And the carbs have a lot of benefit for you. You know, as we get older, we want to clear out more, but we also want to sustain muscle mass, which is mTOR forward. So this is what he was asking. Can mTOR switch on a dime? You could be mTOR blocked in the morning and mTOR forward at night. And this enables you to toggle back and forth. Carbs also make you sleep better at night. So there's your answer.
That's great. I'm going to kind of summarize some of these things. I know that we're running out of time, everybody, but we can get your questions answered, even if I can't answer all of them right now. NAD Platinum is the brand new product, and we have that here. Comes in this type of container. And the big difference is it's taking NAD Gold Plus and adding quercetin, B vitamins.
Yeah, the methyl charge. So it's the NAD Gold, the methyl charge compounds, and then some sirtuin activators, or resveratrol, quercetin.
That's great. And let's see.
So the thing is, you can't individually change them. Sometimes when I'm on, if I'm doing exosomes, I want more. If I want more SAMe, and that's either I'm in a growth phase, or maybe I've been drinking some wine the night before, then I can toggle up the methyl charge separate from the other stuff. So it's nice to have all three, but if you just want a single go-to, it's the NAD Platinum. And for big chunks of my life, it's just NAD Platinum and AMPK charge.
Great. And one of my favorite doctors, Dr. Lay, yes, we were recording this. I know there's a lot of information all in once, and you can review this with us, and we'll have this for you just in a matter of a couple days here. She also asked, are there any medications patients are on that we need to be aware of when we do the detox, like the push catch?
It's hard to go right into specifics on that. Obviously you need to separate the chart, the binder from their medications by a good amount of time. Mm-hmm. But you do have to monitor, if you're doing like an anticoagulant or a blood pressure medicine. Often, like blood pressure can be driven by a combination of AMPK problems or toxins. And if you start washing those out, then all of a sudden your blood pressure is going to start marching down because you don't need the blood pressure med anymore. So watch for the clinical aspects, with anticoagulants, you can watch INR, and just what you truly may have to adjust dosage schedules. But we don't have, I can't tell you right now, oh, this one interacts with that. But, realize you're increasing detoxification. You could be decreasing. So it could go both ways. You could be detoxing the pharmaceutical, or you could be removing the problem that it's compensating for. So you just as a practitioner have to watch that. Now, the nice thing is most of these compounds in the liver sauce go in, do their thing, and go away. So if you're not taking the drug before you do the detox, that detox isn't going to affect the levels of the drug.
Right. And how I understand it is when you take liver sauce, you have about 30 minutes later is ideal to take Ultrabinder.
Yeah, and then about 30 minutes later to eat. Okay. Now, if you're going to take a drug, I'd wait at least an hour, hour and a half. Now, the thing is, the compounds that are stimulating detoxification, like lipoic acid, is gone in 90 minutes. Quercetin is gone in 90 minutes. Most of them are activate, bind, and they're gone. So then when you take the pharmaceutical, an hour and a half, 2 hours later, that biochemistry of the liver sauce is not interfering in it.
Now, if I'm taking liver sauce, would I always be using an Ultrabinder?
Now, you don't always have to. It's especially in the beginning you do. But one of the things you could do, you take it a half hour before you eat, and once you don't have a ton of toxins coming out, your food, especially if you have a high fiber diet, will be the binder.
Ah, that's great. Well, I just want to thank you. That's all the time we really have for all these questions. I hope we got to so many of them, and they're great questions. And I really
appreciate all the questions. It shows real interest and engagement, and that they're taking this stuff, which is fantastic.
We all are, on our patients and ourselves. So it's been such a great journey to be able to have this incredible tool available to us. And so thank you for making these, and also your time today, Dr. Shade. It's a real pleasure. So we will have the content of this presentation available to practitioners only.